Yeah.
So I was like writing out my stuff on like the optical density stuff. So I know like the more cloudy it is, the more dense, obviously.
Is this for lab stuff?
No, for the exam stuff. So like less light's passing through because it's more dense.
Yeah.
But would that mean the absorbance number would be higher or lower?
Yeah, it would be higher. So it does the math and it converts it. And so if less light is passing because it's more cloudy, it means there's more cells in there. So the light and the absorbance.
Oh, so the cells are absorbing more.
Yeah, it's just the math. You don't have to understand like the actual like. I don't even know exactly like the math that goes into it, but it is inversely proportional. Where if less light's able to pass through, you'll have a higher absorbance.
Okay.
So yeah. So yeah, basically the light is absorbing more cells. Actually.
Yeah.
Where she do? Unfortunately, it's in my door. I still have time. I can go get it right now, or I can get it to you after class, whichever.
That's okay. I guess because the purpose of this is just for us to actually like review the concepts and stuff. So if you've completed it, that's totally fine. I'm not gonna collect it or anything. It's just for you. So, do you have look at your computer because I have it on Blackboard.
I have the physical copy, and I filled it all out. All the questions are done.
But that's at home?
That's
Okay, but just for you to have something to like write notes on here, actually, I have another copy of it. So you can kind of follow along and use this today, and then, yeah, and don't worry, like, as long as you've done it, it's really like, you know.
Okay, so I can send you pictures of like, cause I don't. It won't happen again.
It's fine, yeah, this is literally just for you guys to ask me questions, know the concept, the answers and stuff. So.
Yeah, I think it's a really good review. Thank you so much.
Yeah.
After like dinner.
I think Sam's going for Ken, but
Oh, do I have a comment there?
yeah, I've got it. But that's not yeah, I was gonna say. I think today I'd be at the speaker like 10 hours.
Tell me what 2 days will do.
Alright, how's everyone doing today? Anyone do anything fun over the weekend? Anyone study over the weekend? Yeah. Alright, so today, the purpose of today's lecture is for me not to throw more content at you, but for us to kind of take a step back and allow you guys to answer or ask me questions on any of the lectures. So I have them all up here. We can start by going through the answers to the second worksheet. And then, yeah, I have all the lectures up, so if you guys need anything to be explained again or any concepts reiterated, I'm happy to do that with you. So, all right, does everybody have the worksheet with them? Okay, cool. So let's just go ahead and start, and I'm just gonna ask, each one and have you guys answer them. I just ask that not the same people answer all the questions. I kind of want to get a little bit more engagement from the rest of you guys, especially because this is for your own good, it's for the exam, so I want to make sure that you guys are really grasping things and not just depending on those that happily like to answer the questions. So, how is DNA organized in prokaryotes versus eukaryotes? Yeah.
In prokaryotes, it's divided in nucleus, and in prokaryotes, it's a nucleotide that extends to the cytoplasm and domain.
Yeah. Sorry, did you say the first part you said again?
In eukaryotes.
Yeah. Okay, just making sure. I thought I heard you the opposite, but yeah, exactly. Yeah, in prokaryotes, the DNA is organized in a nucleoid, and that's it, and it just is in the same space as everything in the cytoplasm. And then in eukaryotes, it's enclosed in a nucleus. So, all right. Number 2, how and where are the processes of transcription and translation coordinated in a bacterial cell? Yeah.
It says that the main difference, like of them being coordinated, would be that the RNA is immediately bound to the ribosome instead of being translated to separate processes.
Yep, exactly. So that's definitely part of it. So in prokaryotes, or in a bacterial cell, the processes of transcription and translation are coupled, both spatially and temporally, so in the same space and at the same time. And so just to kind of reiterate that, you have the RNA polymerase that binds the DNA, and it runs along and it starts making that mRNA transcript, and that's transcription. And so as soon as this mRNA transcript is made, the ribosomes are there in the same place, so exactly what you were saying, so they can immediately jump on the mRNA transcript, even while that transcript is still being transcribed. And so the ribosome is already jumped up and it's starting translation and making that polypeptide sequence for the proteins. So it's this coordination of the replication of the chromosome and then transcription and translation that helps us understand why bacteria can replicate and divide so quickly. And so the reason is this high coordination of these essential processes. So it's important to kind of know those things.
Is that in the cytoplasm? It just happens.
Yeah, it's all happening in the same space. Okay, any questions on that? All right. So what direction do the replication forks move in when DNA replication begins at the origin And how many DNA polymerases do these replication forks each have? Yes.
There's two replication forks that move outwards, like away from each other, and then each fork has its own DNA.
So yeah, the two replication forks, they move bidirectionally, and then each replication fork has two DNA polymerases. So for the two replication forks, there's 4 DNA polymerases total. Each one has 2. All right. What is the process or the step that completes the cell division, and what's required for the formation of the step?
Is that the septation step, and then you need to replicate the terminator site or the T on it?
Yep, exactly. Yep. So septation is the step that really like separates the 2 cells from each other, and you get this creation of 2 daughter cells. But this septation step can't really happen. The septum can't form until you have the termination site replicated. So you go through the entire replication process, then the septum forms, and they can divide. Okay. Explain the specialized structures, pillized stalks, and nanotubes, and what are some of their key differences? Yeah.
I said villi are straight filaments used in attachment. Stalks are membrane-embedded extensions of the cytoplasm that attach to surfaces. Nanotubes are intercellular connections that pass material from one cell to the next.
Yeah, exactly. So those are all specialized structures of bacteria, and it's just important to remember, what each of those does, for other cells or for itself. What are they used to attach to each other, to surfaces, to other cells, and kind of, yeah, exactly. I have the exact same thing written down. All right, any questions on those so far? What are the 4 classes of organelles that bacteria have? This was more like a memorization question, just something for you guys to know. You don't have to know the details of them, just kind of what they are, the classes. Yeah.
Membrane bound, protein bound, lipid monolayer.
Yeah. Yep, so the key there was that bacteria do have organelles, and so they can be either membrane or protein bound, and the lipid monolayer is defined. We're not going to go into the details of that, but it's just important to know that there is some coordination within bacterial cells, because for the longest time we thought that there were absolutely no organelles. They're not as extensive as eukaryotes, but they do exist, and they come in these different forms. All right, number 7. What are 2 things that can happen when resources or nutrients deplete for bacteria? Yeah.
They can cannibalize or hibernate.
Yep, those are 2 things. What are some others? I know I have 4 on the list for you in the lecture. Yeah.
They could die. They can also die.
Yeah. So which one do you think would be the most common to happen? Die, yeah. So the first thing usually when resources deplete or nutrients deplete for bacteria, usually they'll die, but some other times they can cannibalize others and start to utilize others around them. Kind of natural selection plays in there. When they kind of hibernate and hunker down and wait, how do they do that? What do they form? What allows them to kind of go latent like that? Yep, so they can create some spores, and that kind of is a mechanism that bacteria use, also fungi use to hunker down and wait. They can also kind of start searching a little bit, and there are some specialized structures that allow them to kind of search around for some other nutrients. So what would they need to have to be able to do that? Yep, some sort of motility device like the flagella. And then they can mutate and evolve. So do you guys think that happens often? Yeah, that's really the rare one that would happen, because they need to acquire the perfect mutation to be able to evolve, but it does happen. That's why we still have bacteria around for all centuries that we've had them, and also why there's so much antibiotic resistance. All right, Any questions on that? So what processes would need to occur since we can't use nitrogen gas in its atmospheric form, and who does this? So nitrogen gas, N2, in the atmosphere, like I said, it has it's two nitrogen molecules that are bonded by this really strong triple bond. And so we're unable to use it because of that triple bond. So in order for nitrogen to be usable, something needs to happen. So what would that be? Yep. So nitrogen fixation would need to happen. And what nitrogen fixation is the nitrogen gas is converted into ammonium ions that are usable. And so who does this? What kind of bacteria? Yeah, but yeah, rhizobium is an example exactly of nitrogen fixing bacteria. So one of the experiments in the lab is something that I'm gonna, hopefully, we're gonna be able to do is we'll get some soil and we'll try to isolate some nitrogen fixing bacteria. So that would be cool for you guys to really understand this kind of concept. All right, so explain passive transport and active transport in terms of concentration gradients. What's the difference? And give an example of each. So passive transport, what happens in terms of the concentration gradients?
You go from a high to a low concentration gradient, which means you go from energy to.
Yep, exactly. So passive, the solutes are moving from high to low concentration, no energy is required. Active transport.
With a high, so you done.
Yep, so you need energy. And so so the difference between them is, the difference in requiring energy. So what are some examples of both that I mentioned? Lecture.
An example of passive transport would be facilitated.
Yep, exactly. And then more specifically, like in E. coli, they have this glycerol transport that facilitates the transport of glycerol. So that E. coli glycerol transporter is one of those like more specific examples. An active transport, you don't really have to know like too much detail, but remember the symport and the active or the antiports. So it's important to just understand those mechanisms. If you guys want me to go over that again, I can totally pull up the slide and do that. But yeah, so when they're going from a low to high concentration, you're going to require energy, so they're going against their concentration gradient. And ABC transporters, what needs to occur in order for the membrane transporter to open its channel and release the nutrient molecule? This is one of the key steps that allows the the molecule to be released that is trying to transport.
Is that like the ATP hydrolysis? So you have ATP to go into ADP plus phosphorus?
Yeah, exactly. So in order for the entire reaction to occur and for it to be able to transfer the nutrient through the ATP transporter, you need to have ATP hydrolysis and that conformational change, and that will open up the site and allow it to be released. So we talked a little bit about both liquid and solid media being able to be used to grow bacteria. And so when would you prefer to use solid media over liquid media? When would you rather use a plate to plate bacteria versus having a tube of broth? Yeah.
When you're trying to separate mixed cultures.
Exactly, yeah. So, and if you're plating, you're able to isolate individual colonies. If you have a mixed culture, you can have two different types of colonies growing on some rich media, and that way you can see the differences in either their morphology or their color, that kind of thing. And so for a dilution streak, what's the ultimate purpose of doing a dilution streak? So streak plating. What are you trying to achieve here? Yeah. Yep, exactly. So you're basically creating a serial dilution on a plate in order to, one, isolate individual colonies, but also so that you can get a countable number of colonies so that you can work backwards to calculate that initial titer concentration. So if I give you a tube of some liquid broth and tell you there's an unknown number of bacteria in here, but there's a lot. And so what we can do is do a dilution streak and then end up streaking at, we dilute it several times and then we streak, and then we can count the number of colonies that are on the plate and then use the dilution factor to like work backwards. And so you don't have to necessarily know the math there, but we kind of did a small example last time in lecture. So yeah, you're not only achieving a countable number of colonies, but also you're able to then, you're not only isolating colonies, but you're trying to achieve a countable number of colonies so that you can find the initial titer. Okay, what is the difference between selective and differential media?
Selective favors growth of one organism over another, and differential exploits differences between two species that grow equally.
Yep. So, yeah, exactly. So selective will help one organism grow and doesn't allow the growth of another organism. So you're selecting for just that one type of bacteria. The example that I gave was a gram-positive media or a gram-negative media that allows gram-negative bacteria to grow, but not gram-positives. And then differential is exactly what you said, allows both two organisms to grow that it's selecting for. So it could be gram-negative cells, but there's two different types, and so they can grow differently because the media is differential in that it basically utilizes something that one of those bacteria can do, like maybe metabolize a substrate or something inside the media and then change color. And so color change is an example of one of those things that it can differentiate between. All right, does that make sense? What is a disadvantage of using a hemocytometer to count cells? Yeah, definitely error prone. Counting cells, you're not gonna, be able to very accurately count. Normally, we have like a little clicker thing that you click as you count, and it helps you kind of keep track of the number, but it also is like not the most accurate. Another error that a disadvantage of using like hemocytometers is that in a hemocytometer, you're putting your cells on to that hemocytometer grid, so it's like a little slide and it has that grid inherent in it, but it accounts for both live and dead cells, so you aren't able to differentiate between live and dead cells, so that's one of those things to remember. There's certain dyes that you can use or stains that you can use that will only stain dead cells or that will only stain live cells, but just general hemocytometer counting, you won't be able to distinguish them. All right, so what are biofilms and what's their extracellular matrix or ECM composed of?
Two groups of bacteria that adhere to each other and to a surface. Those called curli, which are protein fibers, cellulose, which are glucose fibers.
Yeah, exactly. Yeah. So biofilms are specialized surface-attached communities of bacteria. And so, yeah, they're composed of these fibers, curli and cellulose fibers. And exactly, curli are protein fibers, cellulose are sugar or glucose fibers. All right. What are some cues that can trigger biofilm formation?
pH, iron, concentration, temperature, oxygen, and then
Yep, exactly. Different things can trigger biofilm formation, and then once they're happy, there can be, just like, you know, I've said before, bacteria go from feast to famine really quickly, and so they can feast and grow and form these biofilms, and then as soon as nutrients deplete, they can start to detach individually and start to disperse, and so you have that whole cycle again. So there's a lot of different things that can trigger their formation and even their falling apart. All right, any questions so far on these?
About like, I'm a little confused about like pili. So I know the stalks are like extensions of cytoplasms, and the nanotubes connect like between each other. But like, what are pili connecting to?
Yeah, so let's go back to that lecture. Great question. Okay. We can go to the next one. So they basically are straight filaments of protein that are just connected to the cell. So they're actually connected to the cell. And they're made of this peelin protein. And so they're attaching to like surfaces here. And so then there are stalks. So stalks are over here, and they are also attached to the cells. So here, like the pili are just really, really thin, like straight filaments, as you can see here, and there could be like many of them that are, you know, hair-like, they're really like hair-like structures of, protein that just kind of come off from the cell and help it attach, and they're really, really thin. Whereas these stalks are these, you know, extensions of the membrane. They're not like an additional like filament. It's literally like the membrane embedded proteins in there are extended, and so it basically helps to attach the cytoplasm to a surface through these stalks. And so one thing that they form is these secretion factors, these holdfasts that are like this glue, and so they secrete this adhesion factor to help it actually adhere to attach to surfaces. And then, so they're membrane embedded. That's the big key difference there. Whereas pili are just straight filaments, like hair-like structures. And then the nanotubes just are intercellular connections. So they connect cell to cell, and they can pass material that way. Does that make sense? Okay. Yeah, so I guess at this point, I know that some of these questions are straightforward. The key for me to kind of get across to you guys is it's as long as you guys are going through the lectures and, you know, there's a little bit of memorization, a little bit of conceptual stuff for like the exam. So if you're able to, you know, go through the lectures and answer these, you should be okay for the exam. And then, but the exam isn't just limited to what was on these worksheets. Anything that I talked about in lecture is fair game. I tried to keep it very straightforward. But yeah, just to kind of give you guys an idea. And then for the rest of this time, I guess I can open it up to you guys to ask me questions on anything that you need clarification on. Yeah.
I just have a question about the exam. What's the format?
It's going to be 50 multiple choice questions. So it'll be 100 points total. You'll have the full 75 minutes to complete it. And so yeah, it'll be Scantron based. So just bring a pencil and that kind of thing. So. Any other questions on any of the content? Yeah.
So I know you talked about like the math for, I think it was the magnification stuff.
Mm-hmm.
Should we be able to like calculate that, or just like?
No, I'm not gonna ask you guys to calculate anything. Yeah, but that would be important for lab because we will be doing that. So things like that I can incorporate into lab, I'm not incorporating into the exam as much because there's a lot of material that I could ask questions about otherwise. Yeah.
Can you go over the ABC system?
Yeah, absolutely. So, ABC transporters are active transporters. And so it's important to know that they are the largest family of transport systems, and they're found in all domains of life. And so this stands for the ATP binding cassette. So this is this ATP binding cassette right here in green. And so the way that this works is so this is a gram-negative cell that I'm showing you here, just for context. So you have the outer membrane, you have the periplasm, and you have the inner cell membrane right here. In the inner cell membrane, you have this membrane transporter, the ATP binding cassette, and you have these two dimers right here. And so this is embedded in the cell membrane, so it's a membrane-embedded protein. And so in an ABC transport system, you have a substrate, so you have this red molecule here, and so this is important, and this is what we want to get through this ATP transporter. So it comes in through a porin, so it comes in through something else, because there's another membrane here, and it can't just pass through, so there's a porin there to let it in. It comes in, and then there is this solute binding protein. So this molecule is the solute, and so there's a binding protein that will bring it in to this next membrane-embedded protein, the membrane transporter. And so the solute will bring it here, and once it does that, it activates this to open up and bind the solute. So this results in this conformational change, and this can get, the solute or this molecule can get embedded in here. And now it's got to get to the other side. And so in order for that to happen, you need, when the conformational change happens, you have these two dimer molecules, they attach. And so once they attach, that activates ATP hydrolysis, so ATP can hydrolyze, and then it can open up again and release the solute into the cytoplasm. So in order for ATP hydrolysis to happen, you need this conformational change where the two dimers actually connect. And then in order for the solute to be able to go through into the cytoplasm, you need this ATP hydrolysis to happen. And so there are different ways that these are used, so that's really the mechanism that I'm explaining, but it's one of the main reasons that these are found in so many different domains of life is it's a critical way to transport nutrients like sugars and amino acids into the cell. But then, like I said, certain bacteria have evolved mechanisms to pump things out that they don't want. So if we're trying to target an ATP binding cassette to maybe get an antibiotic in, for instance, there's ways that they learn to basically pump it out, and so it can be used as like a multi-drug efflux pump. So they're used to basically import things and export things out of the cell. Does that help? What else?
Is there just like a good way to study like the details between like protists and amoeba and fungi, like all those small like categories of things?
Yeah, that part's always.
I feel like I just like am memorizing it, but then I get like confused with like the details.
Yeah, exactly. So I think memorizing those is okay, but there are some key things. Let's see. Oh yeah. It's important to know, are they prokaryotes or eukaryotes? So in archaea, I think memorizing and just really getting these down is important for this. So basically, archaea, I've said, are understudied. We really don't know much about them. And for the longest time, we thought that they just looked like weird bacteria, weirdly shaped bacteria. But then once we've started to have some more techniques to look at them better and understand them better, we were starting to see that, yeah, maybe they look morphologically like bacteria, but there are actual other differences that resemble eukaryotes more so. And so those things are, differences in their cell walls, their membranes, gene expression machinery. So that's archaea. You don't have to really know much about these, different types, but just know that they are extreme organisms, so they're able to really survive many different conditions. For protists, So these are very diverse, and so this one could get a little bit convoluted. So it's important to know that algae are like plant-like, and so they can photosynthesize. And so if you think of green algae, green, you can think photosynthesis. And so they can come in both unicellular and multicellular forms. Where are they found? That kind of thing. Amoebas, they're unicellular, and some slime molds are just fungus-like that form spores. So kind of memorization there. And then within the algae, I have a little bit more detail here, just kind of reiterating the same things, where they're found, what they need to grow. And then I talk a little bit more about phytoplankton, which are photosynthetic organisms within our oceans. What are they important? What kind of processes do? They are really central to the carbon fixing cycle. And so, what do they really offer us? And so phytoplankton provide about 80% of the planet's atmospheric oxygen. And they're really diverse, so you don't have to know these names, the four abundant types, but just kind of understand that microalgae and cyanobacteria fall under this phytoplankton category. And then, what are cyanobacteria? And so that kind of thing. Just kind of memorizing, knowing these details is key here. Amoebas are, like I said, animal-like protists, and so they really lack, they're very fluid-like, because they lack a cell wall and an exoskeletal structure, and so their cytoplasm just flows. And I showed you an example here of an amoeba eating a heliozoan, just kind of getting its nutrients and then going off to do its own thing. So that's kind of what I would expect for that. I think I saw another hand up earlier. Any other questions? Yeah.
Can you just go over biofilms again?
Yeah. Yeah, I kind of ran through the last few slides last week, so I'm happy to go through anything. Bacteria, they exhibit group behaviors, and so that biofilms really help them to resist environmental stressors. So when you have a lot of them together, they kind of, are a pack, and so they adhere to each other and to other surfaces. Through this formation of a biofilm, they can come in many different forms and shapes and stuff. And so this happens in nature, and so there are these just surface attachments, they just attach to each other. They can be one single species of bacteria, or it can be multiple. And so they form, and then when there's a lot of nutrients, because they obviously are thriving on it, and they're able to, get together and form these attachments. But then when nutrients become scarce, they can start to detach and to forage for new nutrients, and it falls apart. So they're constantly going between one and the other. Biofilms are good ways for bacteria to survive and not, get killed by certain environmental stressors. But they also come in the form of here, where plaque on teeth. If you don't brush your teeth often, all of the food and the carbohydrates is a source for these bacteria to attach to the surface, this mucosal surface, and form a biofilm. And so that's one way that they can present. And so what are they held by? They are held by an ECM, or an extracellular matrix. So they are all attached to each other, but then they have to also have a covering that resists denaturation or breakdown from the environment. And so that ECM is composed of these fibers. So so they're called Curli fibers. These are the protein fibers, and then cellulose are the glucose fibers that just really come together to form this tough extracellular matrix, and it helps to resist, denaturation. So like I said, they take different forms and serve different functions. When there's a lot of nutrients, they kind of just sit and are happy. They, help to shelter themselves from, harsh environments or antibiotic treatments. And they also do that to ward off predators and prevent phagocytosis, which is a process in our immune system where, our immune system cells, like macrophages, will try to kill bacteria that's not supposed to be there. And so they do this, they make these biofilms to, get big and strong and prevent being killed off. And, like we said, there's so many different cues. But let's go through a little bit of the stages of biofilm development. So you have these planktonic cells, which are just these individual cells. And so there's a monolayer. Here they have these flagella. And so they kind of are able to swim and, attach to this monolayer surface. And then they start to secrete certain factors. So this is where the quorum sensing comes in, where they it's a communication system for bacteria, where they start to secrete things and can use those cues to know, oh, there's a bunch of us kind of accumulating in this area now. So they all go there. And then you start to get these microcolonies. So these, smaller colonies of these attachments. And then once you have enough of them and, they call in their friends, you start to get this EPS production, which is this polysaccharide. It's just this glue that kind of helps glue them together. And then that brings in more cells. And then you have really this mature biofilm with towers coming off of the center. And so, it's maintained when it matures. And so it's maintained until nutrients start to deplete. And so once nutrients aren't there anymore, they're going to start to need to survive because they're using up everything that they have there. And so that's when they start to dissolve. Then they start to, detach one by one. And then once they lose that really strong connection, they all start to go off and do their own thing. And the process can happen again. So that's biofilms. That's what you should know about biofilms. Does that help? Yes.
I was just wondering, are phytoplankton classified as prokaryotes or eukaryotes, or both?
I don't have anything on here about them being either, so I want to say they can be both, but I'm not going to ask you that. And I will double check just to make sure that I'm not wrong. But let me double check because they come in these two. So the cyanobacteria are prokaryotes. Algae, I think, can be either, but I would lean towards saying they could be both, and I'll double check on that. But I don't have it in here because they can get confusing. There's different types of phytoplankton. So yeah, I wouldn't worry about that. Probably mostly prokaryotes, though. Yeah.
Can you refresh the peptoglycan, the changes it does with mechanisms?
The peptidoglycan, which part?
I guess just a little summary of the different ways it bind or not bind.
Sure. Like the peptidoglycan layer. Is that what you're talking about?
Like with the antibiotics.
Oh, the antibiotic stuff. Yeah. Sure. So, yeah, like we've talked about, the cell walls of bacteria are made up of peptidoglycan. And so one of the key differences between gram-positive and gram-negatives is the thickness of this peptidoglycan layer. So this peptidoglycan layer, it consists of these two sugars, NAG and NAM, And so these are the sugars, they kind of connect in this way and they form these chains. And then, so that's this part of the cell. So that's kind of zooming into the cell wall. So you have these chains that are on both ends, so they're on the top and the bottom, and they have to be linked somehow. And so they're linked by these amino acids. And then these amino acids then form these cross bridges to one another that connect the parallel strands. So beyond this peptidoglycan layer also needs one more step to be able to be clicked together and be strong, and that's having this transpeptidase that helps to cross-link the entire peptidoglycan strands. So there's a lot of different things that keep it intact that go into play. And so basically, since bacteria have these cell walls, you know, when we have a bacterial infection, we want to use antibiotics to kill it. We want to make an antibiotic that will target just the bacteria and not our own cells. And so we take advantage of this peptidoglycan layer within their cell wall because we don't have cell walls, we don't have peptidoglycan. And so we are able to then play around and form antibiotics that can interrupt this cross-linking or interrupt this cell wall formation, because if the cell wall of the bacteria is cracked open, your bacteria are just done, they're dead. You know, when the cell wall is cracked, they're not going to be contained, and you know, they're not going to be able to do what they need to do. They need that cell wall for protection. So if we interfere with that, they're going to die. And so that's where this type of antibiotics comes in. So this peptidoglycan synthesis is a target for antibiotics. These specific types of antibiotics are called beta-lactam antibiotics. These are the ones that target the cell wall synthesis. There are antibiotics that target RNA and protein synthesis and other things, but I'm only talking about the ones that target the cell wall here. So those are beta-lactam antibiotics. And so we target this transpeptidase enzyme that's cross-linking these peptides. And so the way that we do this is these beta-lactam antibiotics bind to the transpeptidase. So all beta-lactam antibiotics have a beta-lactam ring. The mother of the beta-lactam antibiotics is penicillin. It was the first beta-lactam antibiotic to be discovered, and we now have variations of it. So if you hear about amoxicillin, ampicillin, anything that ends with cillin is a penicillin type of antibiotic, but the family is penicillin. So that's why I have penicillin written down here. But the point here is that it has a beta-lactam ring. And so this beta-lactam ring here kind of is shaped like a house in just the way I think of it. And so that's the core part of these antibiotics. And so this is able to go in and bind the transpeptidase. And so if it binds to the transpeptidase, then the transpeptidase can't do its job and cross-link these amino acids and these sugars. So it disrupts this cross-linking, and then the cell wall isn't intact anymore, so water can rush into the cell and it can lyse and burst, and so that's what happens. Now, ultimately, yeah, so having all these antibiotics selects for antibiotic resistance. And so there are ways that bacteria have evolved to be smart enough to resist being killed by our antibiotics. One of those ways is it can create a mutation in this transpeptidase, so it knows that we're trying to actually target this and kill it, so it changes its shape, but it can still cross-link the amino acids, and it can still do its job, but now the beta-lactam ring cannot bind, and so that's one way it can evade death by these mutations in their transpeptidase. Or another way is there's a lot of genetic material shared among bacteria since they all exist in the same environments. And so this trait allows certain genes to jump ship between one species and the next. So there's a lot of genes that are encoded in other bacteria, like this beta-lactamase enzyme. Basically, it's an enzyme that breaks down beta-lactam. So this can then be shared by useless because the thing doesn't work well. But yeah, so the beta lactamase can then be shared here where this transpeptidase was, and so now this cell, this is the DNA, is gonna express an enzyme that's gonna come out and be a beta lactamase. So it's this Pac-Man molecule because it helps to break things down, and so yeah, so that will then target the beta lactam ring, and it'll, you know, break it open so that this beta lactam ring can no longer bind to the transpeptidase. And so the transpeptidase here isn't a different shape, but this just cannot bind here anymore. Those are the two ways of mechanisms of resistance. Does that help? What else? You guys have a good understanding of the cell wall differences and gram positives and gram negatives? Yeah.
I get kind of confused with the gram negative and gram positive.
Okay, yeah, we can talk about it. So gram positive cell envelope, you have the cell membrane, and then you have the peptidoglycan layer, so it's just another way of showing it. So these are the NAG and NAM that I just showed you. And then in between you have these amino acids. But then you also have these teichoic acids that are threaded into the peptidoglycan layers. And so these are negatively charged, and so basically they're important for the gram stain because the gram stain, the crystal violet is positively charged, and so it helps it to really click in there. So that's just an additional thing that the gram negative cells don't have. And so there is this S layer or a capsule that is just this really, like, it's made up of some polysaccharides and proteins, and this just helps it prevent it from phagocytosis. So also these glycosyl chains as well. This all just helps protect it from being eaten up by our immune cells and killed by immune cells. So that's just an additional part that these gram positive cells have. They help keep it sticky and not dry out. The key here is that in gram negative cells, we have a thin peptidoglycan layer, but because it's so thin, it's sandwiched in between two membranes. So here in the gram positives, you have the cell membrane, but then in the gram negatives, you have the cell membrane as well. It's called the inner membrane because then there's an outer membrane. But because there isn't all those different layers of peptidoglycan in here to connect it, there's these lipoproteins that are just these combinations of little proteins that are gonna help it adhere to the outer membrane. And then kind of in the example that I was showing you with the ABC transport system, there was a porin molecule, so there can be some embedded proteins and porins in here that help get things in and then it's gotta come through the outer membrane, so there's these porins, and then there's membrane proteins in here as well. And so, yeah, there's this outward facing leaflet over here. So this is just some leaflet that's on the outside of the membranes, and so this contains lipopolysaccharides, or LPS. Don't worry about it being cross-linked by magnesium ions, but that's kind of how they're cross-linked. And then you have, yeah, like these porins, or these large diffusion channels. So that's on the outward leaflet. The inward facing leaflet is this lipoprotein. So that's just connecting it. That's not in these gram positive cells because they only have one membrane. Does that help?
Yeah.
So it's important to know, these terms, and so you might, think you know it, but then when I like ask a question like this, like which is a component of peptidoglycan, you might forget like which one isn't actually part of the peptidoglycan layer. These are all part of the cell wall, so it's important to be able to differentiate like, oh, the peptidoglycan layer itself has NAG and NAM and amino acids and these peptide crosslinks. But the LPS, the lipopolysaccharide, is part of the cell wall, not the peptidoglycan layer. So just kind of keep those things straight. Yeah. Yeah, so these, yeah, so another specialized structure that we talked about is these thylakoids and these carboxysomes that are part of photosynthetic bacteria. So just like plants have a thylakoid membrane, bacteria that are able to photosynthesize also have these thylakoids, which are just these membranes that are extensively folded within the cells. And they're important for photosynthesis. And then there's also these carboxysomes that are so important in carbon dioxide fixation that occur only in bacteria. So plants don't have this, just bacteria, particularly cyanobacteria. I want to say phytoplankton are prokaryotes now that I think about it more. Let's see. Yep, so over here, this is the nucleoid, and so region, and so you have these white parts in here are the so those are where like the DNA is. In this image you're seeing it, and if you were to just like break the cell open, it's actually pretty coordinated and it's nicely like folded, but it's still in the cytoplasm with everything else in there. So here's the overlay photo or imaging that I'm showing you, where the ribosomes in the cytoplasm and the nucleoid are all in the same part of the cell. They're not in different compartments, but they are organized, they're not meshing, overlapping. You have, your nucleoid in red, and you have your ribosomes and other cytoplasmic content in green there. Yeah.
How much of like the limits of detection do we need to know? Like how in depth?
Yeah.
Like the numbers.
Yeah, so the ones I keep repeating is the limit of detection of the human eye. So the resolution of our eyes, you should know that. And then the limit of detection for a light microscope. So that was 200 nanometers. Important to know, the sizes that prokaryotes and eukaryotes range from. So right here, these numbers and this one I think are important, and this one. Do you guys get the microscopy stuff with how light interacts with matter? Kind of some of the differences here, and how the light is actually gonna, be affected when it hits matter. Those. You guys are good on wet mounts and smear stains, the gram stain procedure. It's been nice because that's also corresponded with our lab stuff, so we've been able to really get those concepts down.
Can you just go over like the gram stain for like, I think it was like the iodine complex.
Yeah, absolutely. So, in the gram stain, you have your gram-positive cells, gram-negative cells. They're both colorless over here. You give them crystal violet, and they're both going to turn purple. So if you remember, E. coli is gram-negative, and in our first stain that we did, the simple stain, the first time we did staining in lab, we just stained E. coli cells with crystal violet, and they showed up as purple. And the reason is because usually if you do a gram stain, they're going to be pink, but that's because we only did a simple stain. We only gave them purple. So they're able to be purple. But once you go through the entire gram stain procedure, things change based on the cell wall. So here at this point, crystal violet is retained reversibly in both. So if you were to use decolorizer at this step, or ethanol, and before the iodine, then you would lose the crystal violet from the gram-positive cells. So in order to trap it in the crystal violet cells, we need iodine first, before the ethanol. So iodine is added because it helps to bind the stain really well to gram-positive cells. So in the way that this works is there's those teichoic acids inside of the peptidoglycan layer of gram-positive cells, and the iodine complexes with crystal violet to make it really strongly charged and be able to bind to the teichoic acids within the peptidoglycan layer. So now the iodine complexes with the crystal violet and helps retain the crystal violet stain in gram-positive cells, but it doesn't do anything to the gram-negative cells. This crystal violet remains reversible because they don't have the teichoic acids in the peptidoglycan layer. So now, at this point, we can use the decolorizer and wash with ethanol or isopropanol, some sort of alcohol, and then that will attempt to wash out the crystal violet from the gram-positives, but now it's locked in because of that iodine, whereas in the gram-negatives, the stain is removed when you wash with this decolorizer. And so then, in order to be able to do a differential stain and actually see them as different colors, we add a lighter stain like safranin that will counterstain the gram-negative cells pink, and then the gram-positive cells still are able to absorb this pink dye, but they remain dark purple because purple is darker than pink. So in lab, we saw that some gram-positive cells did look a little pink or had pink in the middle, a little bit just because it's probably an older culture. Their cell wall wasn't as intact, and so that's why sometimes they can look a little bit more pink, but this is essentially like in a perfect world how the gram stain procedure works. So yeah, if you were to do crystal violet and then add ethanol, then you would lose the crystal violet from both. So that's why we do the iodine after the crystal violet. Does that help? What else? Anything else? Right, well, I mean, there are no more questions, you're welcome to, head out a little early, but if you have any more questions, I'm happy to stay and talk through things, be around for a bit.